Phototropism is the growth of organisms in response to a light stimulus. It is most often observed in plants, but can also occur in other organisms such as fungi. The cells on the plant that are farthest from the light have a chemical called ALll that reacts when phototropism occurs. This causes the plant to have elongated cells on the farthest side from the light. Phototropism is one of the many plant tropisms or movements which respond to external stimuli. Growth towards a light source is called positive phototropism, while growth away from light is called negative phototropism. Most plant shoots exhibit positive phototropism, and rearrange their chloroplasts in the leaves to maximize photosynthetic energy and promote growth. Roots usually exhibit negative phototropism, although gravitropism may play a larger role in root behavior and growth. Some vine shoot tips exhibit negative phototropism, which allows them to grow towards dark, solid objects and climb them. The combination of phototropism and gravitropism allow plants to grow in the correct direction. Mechanism: there are several signaling molecules that help the plant determine where the light source is, and this activates several genes, which change the hormone gradients allowing the plant to grow towards the light. The very tip of the plant known as the coleoptile is necessary in light sensing. The middle portion of the coleoptile is the area where the shoot curvature occurs. The Kolodny-Wendt hypothesis, developed in the early 20th century, predicts that in the presence of asymmetric light, ALll will move towards the shaded side and promote elongation of the cells on that side to cause the plant to curve towards the light source. ALLs activate proton pumps, decreasing the pH in the cells on the dark side of the plant. This acidification of the cell wall region activates enzymes known as expansins which break bonds in the cell wall structure, making the cell walls less rigid. In addition, the acidic environment causes disruption of hydrogen bonds in the cellulose that makes up the cell wall. The decrease in cell wall strength causes cells to swell, exerting the mechanical pressure that drives phototropic movement. A second group of genes, PIN genes, have been found to play a major role in phototropism. They are ALll transporters, so it is thought that they are responsible for the polarization of ALL. Specifically PIN3 has been identified as the primary ALL carrier. It is possible that phototropins receive light and inhibit the activity of pinoid kinase, which then promotes the activity of PIN3. This activation of PIN3 leads to asymmetric distribution of ALL, which then leads to asymmetric elongation of cells in the stem. PIN3 mutants had shorter hypocotyls and roots than the wild type, and the same phenotype was seen in plants grown with ALL efflux inhibitors. Using anti-PIN3 immunold labeling, movement of the PIN3 protein was observed. PIN3 is normally localized to the surface of hypocotyl and stem, but is also internalized in the presence of brefeldina, an exocytosis inhibitor. This mechanism allows PIN3 to be repositioned in response to an environmental stimulus. PIN3 and PIN7 proteins were thought to play a role in pulse-induced phototropism. The curvature responses in the PIN3 mutant were reduced significantly, but only slightly reduced in PIN7 mutants. There is some redundancy among PIN1, PIN3, and PIN7, but it is thought that PIN3 plays a greater role in pulse-induced phototropism. There are phototropins that are highly expressed in the upper region of coleoptiles. The two main phototropins are PHOT1 and PHOT2. PHOT2 single mutants have phototropic responses like that of the wild type, but PHOT1 PHOT2 double mutants do not show any phototropic responses. The amounts of PHOT1 and PHOT2 present are different depending on the age of the plant and the intensity of the light. There is a high amount of PHOT2 present in mature Arabidopsis leaves, and this was also seen in rice orthologs. The expression of PHOT1 and PHOT2 changes depending on the presence of blue or red light. There was a downregulation of PHOT1 mRNA in the presence of light, but upregulation of PHOT2 transcript. The levels of mRNA and protein present in the plant were dependent upon the age of the plant. This suggests that the phototropin expression levels change with the maturation of the leaves. Mature leaves contain chloroplasts that are essential in photosynthesis. Chloroplast rearrangement occurs in different light environments to maximize photosynthesis. There are several genes involved in plant phototropism, including the NPH1 and NPL1 gene. They are both involved in chloroplast rearrangement. The NPH1 and NPL1 double mutants were found to have reduced phototropic responses. In fact, the two genes are both redundant in determining the curvature of the stem. 5 models of OIN distribution in phototropism. In 2012, Sakai and Haga outlined how different OIN concentrations could be arising on shaded and lighted side of the stem, giving birth to phototropic response. 5 models in respect to stem phototropism have been proposed, using Arabidopsis thaliana as the study plant. First model. In the first model, incoming light deactivates OIN on the light side of the plant, allowing the shaded part to continue growing and eventually bend the plant over towards the light. Second model. In the second model, light inhibits OIN biosynthesis on the light side of the plant, thus decreasing the concentration of OIN relative to the unaffected side. Third model. In the third model, there is a horizontal flow of OIN from both the light and dark side of the plant. Incoming light causes more OIN to flow from the exposed side to the shaded side, increasing the concentration of OIN on the shaded side and thus more growth occurring. Fourth model. In the fourth model, it shows the plant receiving light to inhibit OIN basimptal down to the exposed side, causing the OIN to only flow down the shaded side. Fifth model. Model 5 encompasses elements of both Model 3 and 4. The main OIN flow in this model comes from the top of the plant vertically down towards the base of the plant, with some of the OIN traveling horizontally from the main OIN flow to both sides of the plant. Receiving light inhibits the horizontal OIN flow from the main vertical OIN flow to the irradiated exposed side. And according to the study by Sakai and Haga, the observed asymmetric OIN distribution and subsequent phototropic response in hypocotyls seems most consistent with this fifth scenario. Effects of wavelength. Phototropism in plants such as Arabidopsis thaliana is directed by blue light receptors called phototropins. Other photosensitive receptors in plants include phytochromes that sense red light and cryptochromes that sense blue light. Different organs of the plant may exhibit different phototropic reactions to different wavelengths of light. Stem tips exhibit positive phototropic reactions to blue light, while root tips exhibit negative phototropic reactions to blue light. Both root tips and most stem tips exhibit positive phototropism to red light. Cryptochromes are photoreceptors that absorb blue/UVA light, and they help control the circadian rhythm in plants and timing of flowering. Phytochromes are photoreceptors that sense red-far red light, but they also absorb blue light. The combination of responses from phytochromes and cryptochromes allow the plant to respond to various kinds of light. Together, phytochromes and cryptochromes inhibit gravitropism in hypocotyls and contribute to phototropism. See also, scotobiology, Uladnar Eurowent model. References. External links, media related to phototropism at Wikimedia Commons, time-lapse films, plants in motion.